our last speaker of this morning. Next is Narasimha Rao, who's a senior scientist of the Global Clinical Management Function of Dr. Reddy's Laboratories Limited. He obtained his master's degree in biotechnology in 2006 from the University of Madras, India, and started a career as a research officer in Virology R&D and vaccine manufacturing company. And he's currently working in Dr. Reddy's, a leading multinational pharmaceutical company based in India. He's been with Dr. Reddy since 2010 and worked on preclinical assessment of high-value therapeutic products and small molecules, drug repositioning, and drug discovery programs. And currently, he's investigating immunogenicity of complex therapeutic generic peptides. And the title of the talk is FDA Recommendations on Immunogenicity Risk Assessment of Synthetic Peptide Drugs and Their Impurities. Go ahead and share your slides. Thank you.
Hello, everyone, and thank you, Emily, for the kind introduction. I'd like to thank ProImmune for extending the invitation to speak in Mastering Immunity 2023 Summit, a platform where knowledge sharing happens to the scientific community. Today, I'll be sharing FDA recommendations on in vitro immunogenicity risk assessment of synthetic peptide drugs and their impurities. In this talk, I will first give a brief introduction on generic synthetic peptide drug products and impurities, then on innate and adaptive immunogenicity. Lastly, I talk on advice of FDA on in vitro innate and adaptive immunogenicity assays for evaluating the risk of these complex peptide products. Before I begin, I would like to make a disclaimer. The information presented herein is my perspective and not necessarily reflective of those of regulatory agencies. Peptide synthesis is a great way to make peptide drugs, and the only problem being that impurities can be generated in the process, and therefore we need to demonstrate that our peptide drug product and its related impurities does not increase safety risk or immunogenicity. So what is an impurity? Any component of the new drug product that is not the drug substance or an excipient in the drug product is known as an impurity. Although many of these impurities can be removed during the final stages of the drug substance purification, still these impurities can end up in the final drug substance and further into the drug product. In the manufacturing process, impurities can derive from any step, like peptide impurities, process impurities, excipients, leachables, other contaminants that may have immunogenic potential. Immune responses to these impurities can significantly affect the safety and efficacy of these peptide products. Thus, assessing the risk of peptide products inducing an immune response prior to its clinical use is very important. What is immunogenicity? It is defined as the propensity of the peptide product to generate immune responses to induce immunological related adverse clinical events. Some of the consequences for safety are like promote allergic reactions on the injection site. The peptide drug can lead to formation of neutralizing antibodies, which are called as anti-drug antibodies, which can diminish efficacy of the peptide drug product. Also, hypersensitivity responses can lead to cytokine release syndrome. The abbreviated new drug application or ANDA pathway recommends that applicant provides information on impurities in the generic drug compared with the originator product as part of the approval process to ensure patient safety. In general, immunogenicity is characterized into innate immunogenicity and adaptive immunogenicity in humans. First, the innate immune response is body's first line and usually rapid defense against the pathogens. The cells involved in the innate immune system are like dendritic cells, macrophages, mast cells, neutrophils, basophils, and others. In the drug product, the process-related impurities such as contaminants can trigger innate immune response leading to local inflammation. The FDA guidance recommends of testing the whole drug product for innate immunogenicity risk of product and process-related impurities. Unlike the innate immune system, the adaptive immune system relies on fewer types of cells that are antigen-specific T cells and B cells, which leads to production of antibodies. So the applicants are asked to investigate whether their peptide-related impurities have T cell epitopes, which can have increased affinity to bind to T cells, leading to undesired immune response. FDA has released the guidance in May 2021 for applicants seeking approval for synthetic peptides on assessing the immunogenicity risk that focuses on 5 complex peptides. For ANDA submission, firstly, applicant needs to show that for each peptide-related impurity that is found in both the proposed generic synthetic peptide and the reference listed drugs, the level of impurity in the proposed synthetic peptide is same as or lower than that found in the reference listed drugs. Also, to show that the proposed generic synthetic peptide does not contain any new specified peptide-related impurity that is more than 0.5% of the drug substance. This information will enable FDA to evaluate the purity of the proposed generic synthetic peptide and to confirm that it and the reference listed drug of recombinant DNA origin can be expected to have the same safety profile when administered to patients. Let us understand the type of impurities found in the generic synthetic peptide drug. First is process and related impurities. These are innate immune response modulating impurities which influence the innate immune system. Careful evaluation and control of these impurities are crucial to ensure safety and efficacy of therapeutic products and to meet regulatory requirements. Second is peptide-related impurities. These are found in the drug substance. The focus is to conduct an in vitro assay for purified impurities and understanding what is the capacity of these individual impurities to bind to MHC and stimulate T cell activation. FDA recommends a non-clinical immunogenicity evaluations comparing a proposed synthetic generic peptide to the reference listed peptide product of recombinant DNA origin. Here are the key FDA recommendations on immunogenicity risk assessment. Applicant needs to provide a comprehensive impurity profile of the proposed drug product and reference listed drugs. A minimum of 3 lots of proposed drug product and 3 lots of reference listed drug product to be assessed for immunogenicity risk. These studies should be conducted on each proposed strength. The proposed drug product should be manufactured from minimum 2 batches of drug substance or API. Applicant should provide a table describing the details of all the samples tested and capture the product age of testing in their study reports. The immunogenicity risk assessment assays should be validated and should detect impurities risk. In vitro immunogenicity assays should be carefully developed and fit for the purpose to provide data that informs immunogenicity risk of the generic peptide product. When coming to FDA advice on in vitro innate immunogenicity assays for assessing innate immune response, innate studies should be conducted at near release of the product and at the end of proposed shelf life comparing both drug product and references to drug. Use of multiple cell lines that express different pattern recognition receptors are acceptable, and these cell lines are commercially available. A minimum of 20 donors to be tested in the Innate assay if using human whole blood assay or human PBMC assay to account for donor variability. The applicant should provide assay conditions such as duration of assay, number of cells per well, concentrations of the drug product, and suitable controls information. Demonstration of assay sensitivity is very critical here. By providing data that assay can detect broad spectrum of potential impurities is needed. The drug product should be minimally manipulated and that does not decrease cell viability. Spike product prior to any manipulation of the drug product and demonstrate signal recovery to establish sensitivity. Applicant should provide a complete assay description and the relevant data to demonstrate that the assay is suitable for its intended purpose. Some of the common deficiencies on Innate assay identified by FDA are not providing a rationale for the selected cytokine readout, insufficient demonstration of assay sensitivity, not providing sufficient details on the methodology, not examining the formulation effects. So by taking all this into consideration and advice for the applicants, they can avoid common deficiencies of innate immune assays. Next, let me share FDA advice on in vitro adaptive assays for assessing adaptive immune response. Adaptive studies to be conducted using at least 3 lots of drug products and 3 lots of reference drug, and also individually identified peptide related impurities. The FDA is curious about characterizing each individual impurity and hence recommends testing them individually for the presence of T cell epitopes leading to immunogenicity. Applicant should assess the immunogenicity risk of each individual impurity separately at the maximal concentration that does not reduce cell viability or impair their metabolic activity. For in vitro adaptive assay, PBMCs can be chosen as they are clinically relevant, and at least 40 donors are needed to account for donor variability. The study report should include information like cell culture conditions, cell preparations, MHC coverage details, suitability controls for naive T cell response and memory T cell responses. Assay should also demonstrate that it's fit for the purpose, and lipopolysaccharide, phytohemagglutinin, or key lymphocyte hemocyanin are not recommended to detect the presence of naive T cells. So some of the common deficiencies on adaptive assays identified by FDA are that applicant did not demonstrate that the assay is suitable or did not demonstrate the selected PBMC population has HLA class diversity, or the assay has insufficient optimization of the conditions such as duration of the assays, number of cells, concentrations of the drug product, suitability controls, etc. Insufficient method validation like assay sensitivity and specificity. With that, I like to conclude my presentation by saying that immunogenicity risk assessment is critical for generic synthetic peptide drug products. The immunogenicity risk of generic synthetic peptide products needs to be comparatively assessed to the reference drug product, which includes both innate and adaptive immune response. Immunogenicity risk assessment strategies may help mitigate risk of unwanted immune responses. FDA recommendations and thoughts are frequently changing to ensure the safety of the complex generic peptide product. Please remember, immunogenicity assays should demonstrate sensitivity and fit for the purpose of assessing immunogenicity risk of the peptide drug product. We have worked closely with ProImmune, and ProImmune has the expertise in providing immunogenicity services and that are best in class to evaluate immunogenicity risk of the complex peptide product. Dr. Reddy's experience with ProImmune is great, and the professionalism they display supported us in filing our peptide products timely to the regulators. Thank you, ProImmune team. Thank you all for your attention.
Great, thank you so much for that. Are there any questions?
Yes,
Hello.
Emily, there's one
Okay.
one from Disha, and there's also a written one in the chat.
Okay, my chat.
So I have a few questions. Am I audible?
Yes.
Yes. So my first question is, when you say this assay should be validated, so can we develop and qualify or validate a common assay for a different peptide, or for each product it should be different assay only?
So you can validate an assay by your suitable controls, nothing but your assay controls, and that
Yes, I
can be used
understood,
for.
but can
Mhm.
there be a single platform method which I can use for multiple products, or valuation should be performed for each and every product?
As on date, there is no single assay that is been standardized for assessing all the peptides. So it's like for every peptide, you need to identify the right assay platform to assess.
Okay, and if we are not using components like LPS or some positive control which we are sure of inducing immunogenicity, then what will be the positive control for the assay? Like how can we assess that the assay is working apart from our product?
If I understood rightly, so you meant to say other than LPS, what should be used? That was your question.
Yes, 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 as a positive control.
Yeah, for innate assays, you can take different receptor agonists, like for TLR, or NLR, or CLR.
Okay,
Yeah, for adaptive assays, the standards that are being used like PPD, CEFT, peptide pools, etc.
okay, and here we are using drug concentration which is optimal and which is not affecting the viability. So how can we extrapolate this results for actual doses which are going into patients? Because there will be definitely some difference.
Yeah, thank you for asking the question. So I want you to treat this assay as very independent assay, and this is nothing to do with the dosage.
Okay.
Agency is looking for you to do your titration in this particular assay, and then show them the results.
Yes, I understood, but at what point we will say that this molecule is immunogenic or not? At what response level?
So, so basically, that's how you titrate your product in the assay, and as you mentioned, saying that whichever is max safe concentration that is not toxic to your cells. So you pick up that
Okay.
concentration and then you perform your assay.
Oh, all right. Thank you.
Okay, great.
And apart
Thank you.
from generic, if there is a novel molecule, then how will we decide that if the molecule will be immunogenic at that particular level or not?
You mean to say if it's a novel molecule altogether?
Yes, novel molecule altogether.
Oh, then it should be done using clinical trials and then dosage of those
Okay.
things, because we are talking about generic peptides now.
Okay. All right. Thank you.
Okay, great. We have a question from Joshi, and it is in vitro immunogenicity still required for complex peptide generics if all impurities matches and within the limits as per guidance in comparison with the reference standard? And there's a follow-on to that as well.
Okay. Thank you, Joshi, for asking the question. So even if all the impurities are similar to the proposed generic product, still agency recommends that you perform the assessment and show them that your product impurities are having that safety profile.
Okay. And if there are new impurities, can they go straight away into the innate responses with pure impurities along with the test and standards without doing adaptive and in silico?
Can I just repeat? Sorry.
Yeah, this is from the chat, again from Joshi, and it says, if we see new, oops, I'm still lost it here.
I can tell him.
Okay, yes, go ahead.
Hi, Narasimha. So if we see new impurities, we know those impurities are like more than what is recommended as per the agencies, like bit higher. Can we straight away go doing innate response rather than doing adaptive and in silico?
So ideally, yes. But again, as you know, that agency wants that the impurities should be assessed in multiple platforms to make sure that in all possibilities it's safe. So still, I would assume that agency would come back and ask they wanted in silico or adaptive on this. But innate would be the strongest assay supporting the safety of the product, definitely.
Thank you.
Professor Choi, would you like to ask your question?
Yes, please. Yeah. But just the quick questions. For the adaptive, the assays, the recommendation is like 40 PBMC donors at least, and you talk about the MHC target population of the MHC. Is there the concrete number recommendations, like how much percentage should be covered or something like that?
Thank you for the question. So FDA is telling it's minimum 40, but the more number, then less variability. So there is no concrete answer for this question.
And it's the one short question. So where can I find the document for your talk? FDA recommendation.
I can only refer to that. Yeah.
I can share the FDA guidance. I'll just put
yes.
that in the chat right now. 1 second. Okay, I'll put that in the chat.
Thank you very much.
Are there any other questions?
Hi all, I have one question. Am I audible?
Yep, go ahead.
I hope. Yeah. It's not limited knowledge in this field because I'm from business team. I wanted to ask one question. If I change the source of my peptide in my finished formulation, which has been tested for immunogenicity and in clinical trials, say from synthetic peptide, I am now shifting my source of API to biological origin. So what type of studies I need to demonstrate to agencies like FDA, whether I need to perform any immunogenicity study particularly, or I need to do clinical trial again? I hope my question is clear.
If you are okay, for that question, I do not have an answer.
Okay, thank you. So anyone from ProImmune team would like to share their thoughts?
Yeah, could you just sorry, could you say the question again just so I can understand it?
My question is, if I change the source of API in my formulation, in peptide formulation, I'm saying, if it is already approved and it's in market, but in between, if I want to change the source of API from synthetic peptide to, I want to shift to biologically synthetic peptide. So in that case, to my formulation, which type of immunogenicity studies I need to demonstrate, whether any immunogenicity studies are sufficient enough to agency, or I need to do some clinical trials with that formulation.
I would suggest that you speak to the agency to seek clarification on that one, because that's going to be something that only they can answer.
Okay, great, thanks.
Yeah, thank you.
Any other questions? Okay, well, thank you again to our first session speakers. Really nice work, everyone.